Hello and welcome to lecture number 7. This lecture we're going to cover inorganic chemistry and organic chemistry. So inorganic chemistry, there's usually no carbon. We have salts and we have many acids and many bases. Where organic chemistry, it contains carbon molecules, and usually they're very large and they form covalent bonds. These would be carbs, lipids, proteins, and nucleic acids. All right, so let's start with inorganic compounds, water. Water is the most abundant compound in all of our bodies. It accounts for 60 to 80% of the volume of all of our living cells. Water declines as we age. So younger people, such as infants, have a larger percentage of water, whereas we age, we lose our water content. We have less percentage of water. It's the most important molecule in our bodies. It has many properties that are very important. High heat capacity, high heat of vaporization, polar solvent properties, cushioning, and reactivity. Let's talk about those. So, the high heat capacity. This is heating water. Water holds heat for a very long time, longer than other molecules. That's why when you put something on the stove, it takes a little while for it to start to boil. It doesn't just get hot instantly. So this is our ability to absorb and release heat with little temperature change. So the high heat capacity prevents sudden changes of temperature, which is very important in our body because we cannot handle large temperature changes. That's why most of the time your body is 98.6. Okay, high heat of vaporization. This means that it takes a lot of heat or energy to break it down from a liquid to a gas. This is why we sweat. Water's on our skin, it evaporates, it pulls heat away from our body, and it cools us down. Next is the polar solvent properties. We know water's polar, and it's very good at dissolving things. The positive end of water pulls on negatively charged things like chloride and salt. The negative side of water pulls on the positively charged things like sodium side of salt. So it rips that salt in half. It is no longer NaCl. Now it's Na+, which is a sodium ion, and Cl-, which is a chlorine ion. And then the water molecules surround those ions. So that's what's happening when you're watching salt dissolve in water at the molecular level. Reactivity. Water is very reactive, and that's because it's polar. Remember, it has the positive side and the negative side. It's pretty much responsible for every reaction that's happening in our body. Cushioning. Water surrounds many of our organs. It is in our cerebrospinal fluid. It cushions our nervous system. It's in our brain. It cushions our skull, our brain inside of our skull. So it is responsible for cushioning in our bodies. Okay, so this slide, it's demonstrating dehydration synthesis and hydrolysis. So the first box up here is dehydration synthesis. The second one is hydrolysis. So we know that biological molecules are formed from their monomers or units by dehydration synthesis and broken down into monomers by hydrolysis reactions. So in this situation right here, dehydration synthesis, monomers are joined by removing a hydroxyl group. symbolized by the OH, from one monomer, and removing a hydrogen ion from the other monomer. This creates a water that is released from those. So one hydrogen in the OH group, that's going to tear off of those and release a water molecule. Then the oxygen is going to bind over here, forming just the oxygen in the middle. So dehydration synthesis removes a water molecule at the bonding site and results in two monomers being linked by a covalent bond with an oxygen. All right, part B. In hydrolysis, so remember hydro means water, lysis means to cut. So monomers, two monomers that are attached, are released. So they're going to break these apart by adding a water molecule. So when you add the water, that's going to break this bond, and an OH group is going to attach to one monomer, and the H is going to attach to the oxygen to make it its own monomer again. So dehydration synthesis and hydrolysis is the reverse. Okay, so we have two monomers giving off a water, resulting in one monomer that's linked by covalent bond. In hydrolysis, we have a monomer linked by covalent bond. When you add a water to that, it ends up with two separate monomers. So let's talk about acids and bases. These are both electrolytes. They ionize and dissociate in water. Acids are considered proton donors, means they release hydrogen ions. So an example of that would be hydrochloride, HCl. It's going to yield a hydrogen ion and a chlorine ion. So an acid, proton donors, they're going to release hydrogen ions. So that's why we have a hydrogen ion here. So it's going to break apart the H and the Cl into ions. Important acids that we have is hydrochloric acid, acetic acid, and carbonic acid. Bases, these are proton acceptors. They pick up hydrogen ions in solution. So an example of that would be NaOH. It's going to break apart and yield a sodium ion and a hydroxyl ion. So when a base dissolves in a solution, it releases this hydroxyl ion, and that's what's happening. You have the base, dissolves in a solution, it ends up being a sodium ion and a hydroxyl ion. The most important one in our bodies is bicarbonate, and that's HCO3 and ammonia. All right, back to these electrolytes. Electrolytes are actually things that when you put them in water, they develop a charge. So when we talk about acids and bases, we're also talking about pH. Protons, hydrogen ions, and H+, those are all the exact same thing, just different way to write them. And pH is the proton concentration of a solution. In our normal body, pH is 7.35 to 7.45. And we're going to come back to that later. So let's talk about pH. pH is a measurement of the concentration of hydrogen ions in the solution. We know that pH, each unit represents a 10-fold difference. So we're going to talk about that in a minute. So if it's an acid, the pH range is anywhere from 0 to 6.99 on the pH scale. The lower the pH, the more acidic. So as you go from 3 to 2 to 1, that's becoming more acidic, the lower the pH. The lower the pH also means it's increased hydrogen concentration, so more hydrogen ions. And in each step, so going from 4 to 5 has 10 times the hydrogen concentration. So 4 has more hydrogen ions than 5. 5 has more hydrogen ions than 6. 5 has 10 times as much hydrogen concentration as 6. So if I was to ask you, 3 has how much more hydrogen than 5? The answer to that is 3 has 100 times more hydrogen concentration than 5. So it's 10 times each one, each step. So going from 3 to 4, it would be 10 times. But then going from 4 to 5, it would be 10 times that. So that's 100 times. Alkaline solutions or basic solutions. So alkaline range is anything from 7.01 all the way up to 14. So the higher number pH is more alkaline or more basic. The higher the pH means there's less hydrogen concentration. Or you can also say more or increased OH negative concentration. You can do that one either way. And each step of that gives 1/10 times the hydrogen concentration. So an example of that is 9 has 1/10 the hydrogen concentration of a pH 8. Then we have neutral. Neutral have equal numbers of hydrogen and OH ions. They're always going to be around 7, and pure water is pH neutral. Here is the pH scale. So, as we said, neutral is going to be around pH 7. These are going to be things like milk, blood, those are going to be right in there, water. As we move up the pH scale from 8, 9, 10, 11, 12, 13, 14, these become more basic. So this would be a stronger base than oven cleaner. Oven cleaner would be a stronger base than household ammonia. As we get closer to neutral, they get weaker. Likewise, as we go lower on the pH scale, it's going to become more acidic. So black coffee is around a 5, wine is around 2.5 to 3.5, lemon juice, pH of 2, and then hydrochloric acid is going to be way down here at 0. So the more we move lower on the pH scale, the more acidic it is. Okay. So we're going to talk about buffers. When we have a change in our pH, there's a buffer that's going to always bring it back to where it's supposed to be. Because our bodies cannot function well when we're off from our normal pH. So buffers resist abrupt and large swings in pH. They can release hydrogen ions if the pH rises. They can bind to hydrogen ions if the pH falls. It's basically going to bring it back to the normal homeostatic area that it needs to be. Bicarbonate is our most important buffer of our blood. So, Our normal pH is 7.35 to 7.45. We know that it cannot deviate much from that or we're going to not be doing well. Buffers that cause it to come back to its equilibrium is going to be your kidneys, your lungs, and chemicals in your body. All right, organic compounds. So organic molecules contain carbon, with the exceptions of CO2 and CO, which are inorganic. So we just want to make sure that you know that when we talk about carbon, these are going to be forming 4 bonds with other things that are not oxygen in order to be considered organic. Carbon is electrical neutral. It shares electrons. It never gains or loses them. That's why we say it forms covalent bonds. It shares the electrons. And it can form up to 4 covalent bonds. And carbon is unique to living systems. So when we think of carbon, we can make larger compounds called macromolecules. These are just really big molecules. These are in our body, and we're going to learn about them: carbohydrates, lipids, proteins, and nucleic acids. So carbohydrates. Carbohydrates are monosaccharides, simple sugars containing 3 to 7 carbons. Okay, all of these pictures here show monosaccharides. So we have each one of these points here is going to be a carbon. All right, and monosaccharides always contain carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. So like glucose, we know is C6H12O6, it's going to be like that. So we have 6 carbons, 12 hydrogens, okay, if you count them all up, there will be 12, and 6 oxygens. So they always have that general formula of 1 to 2 to 1 ratio. Monomers of carbohydrates are going to be monosaccharides. Important monosaccharides are pentose sugar, ribose and deoxyribose, so here and here. And then hexose sugars is going to be like glucose, which is important in your blood sugar. All right, disaccharides are double sugars. So now we have 2 sugars hooked together. These are too large to pass through cell membranes. The important ones that we know here are sucrose, maltose, and lactose. These are formed by dehydration synthesis of 2 monosaccharides. So we get 2 monosaccharides, like glucose and fructose, from the previous slide, and they bond together to form sucrose and water. Okay, so we're forming that by dehydration. So we're adding those 2 sugars together, and then out with it comes a water. So you're dehydrating it, kind of like taking out a water by bonding these 2 sugars to form a sucrose. Polysaccharides. These are polymers of monosaccharides. So we take a sugar, a sugar, a sugar, a sugar, a sugar, sugar, sugar. We're bonding them together now. It can either be all ones, single sugars, or disaccharides forming with 2 others, but it's just multiple monosaccharides chained together. These are formed by dehydration synthesis. Same thing, it releases water. Important ones on here is going to be carbohydrate storage, starch, glycogen, and these are not very soluble. All right, the next one is lipids. Lipids contain carbon, hydrogen, and oxygen. but less than in carbohydrates, and sometimes they contain a phosphate group. These are very insoluble in water. Just like if you put oil in water, it's not going to mix, it's going to stay separated. They're hydrophobic, means water fearing. Okay, so they do not go well with water. That also means nonpolar. The main types is triglycerides, phospholipids, steroids, and eicosanoids. We're not going to learn too much about eicosanoids, but the other three, yes, we will. All right, lipids. Let's start with triglycerides. These are called fats when they're solid, and oils when they're liquid at room temperature. They're composed of 3 fatty acids, which are linear hydrocarbons. So that just means that they are carbons with hydrogens attached, and they're in a long line. And those are bonded to a glycerol or a sugar alcohol by dehydration synthesis. The main functions of triglycerides is energy storage, more for long term, insulation and protection. So as far as energy storage is concerned, as we learned before, carbs, they're easy to break down, we get quick energy from them. Lipids are difficult to break down, and so those are going to be considered more of a long-term energy. Insulation, this is used because we have fat that's underneath our skin and around our organs, and that it helps to insulate, to keep the body temperature regular, and it helps to cushion our body organs, which acts as protection. Okay, so this is showing how we get the fatty acid. So here we have a fatty acid, which is all these carbon atoms attached to hydrogen ions. Okay, so it just makes a really long fatty acid chain. And then when you put it with a glycerol, you're going to have a dehydration synthesis. So it's going to take the hydrogen ion and the OH from the fatty acid, and it's going to bring out a water, and then those two are going to bond together there to where you have glycerol with a fatty acid. And this can happen for 3 of these. So if you take another fatty acid and bring it up to here to this OH group, it'll throw out another water and it'll bond there to make the second triglyceride. And then if you bring another fatty acid and bond it to this OH, we get another one. So this is a triglyceride. That's why it's tri means 3. Triglyceride. So tri fatty acids, 3 fatty acids, and a glyceride. This is another way to draw it without all the hydrogen ions. It's just, you know that wherever there's a turn, there's a carbon ion there, a carbon atom there. All right, triglycerides can be constructed of saturated fatty acids. So on this, a saturated fatty acid means we have that glycerol here, and we have our fatty acid chains, and these are all straight fatty acid chains. Okay, they're not curved or anything like that, they're just all straight. So all the carbon ion atoms are linked via single covalent bonds, resulting in a molecule with the maximum number of hydrogen ions. And being that it has the maximum number of hydrogen ions, 1 on each side of each carbon, it forms a straight chain. These are packed closely together, forming a solid at room temperature. So example of a saturated fat would be animal fat, lard, butter. These are considered unhealthy. Unsaturated fat. These are 1 or more carbons are linked via a double bond. So that's why you see here the 3 straight fatty acids And then you see this one here, instead of it being nice and straight like these, it has a double bond, which makes it curve down. Okay, it bends the chain. This results in one less, or two less actually, hydrogen ions. So that means it's unsaturated by hydrogen. The double bond forms a kink, so they're not packed so closely together. And this enables water to be able to get in there. So unsaturated is better for you. These are such as olive oil, plant oils. Unsaturated, two other ones that we have is trans fats. These are going to be unsaturated fatty oils that look like saturated. So basically they take an unsaturated, they remove the double bond, and then they make all those fatty acids actually straight again. So what does that give you? Remember that the form is also the function. So once you take the kink out and now they're tightly packed close together again, that's going to be a saturated fat. So that's also going to be unhealthy. So trans fats are also unhealthy, just like lard and bacon grease and all those ones like that. Omega-3 fatty acids, these are considered heart healthy, and these come from fish. All right, the next one is phospholipids. These are modified triglycerides, the glycerol and two fatty acids plus a phosphate group. These are special because they have a head and a tail region, and the head is polar or hydrophilic, it attracts to water, and the tails are fatty acids which are nonpolar and hydrophobic, they repel water. So these have a polar activity where one side can attract water, the other side is repelled by water. And these are going to be very important when we talk about cell membrane structure. So here we have the polar head. Okay, so this is going to be your glycerol, your phosphate, and your nitrogen group with two long-chain fatty acids. Okay, so this one is going to be a double bond here. We see it's bent. So the tail end here is going to be nonpolar, because remember this is a fatty acid. So it's just going to be hydrogens, carbons, and that's it. Up here we have the polar head, which is the hydrophilic group. This is the water loving. So water loving, water fearing. That's the parts of the phospholipid. Here we see the chemical structures. This is the long chain fatty acid, the glycerol, the double bonded fatty acid. Here we have a phosphate group and a nitrogen containing group. So this is kind of an easier way to look at it. We have the polar head, hydrophilic, water loving, and then we have the two fatty acids, the tails, that are water fearing. So when you line them up like this creates a phospholipid bilayer, which is part of our cell membrane. Some other lipids that are important are steroids. These are going to consist of 4 interlocking ring structures. The most important one in our bodies is cholesterol. And, a lot of times this gets a bad rap because if it's too high, but we do need cholesterol. This is made by the liver, also found in animal products such as meat, cheese, eggs. We need cholesterol to synthesize vitamin D, to have steroids, hormones, and bile salts. It's also important in the plasma membrane structure. So here is a steroid structure. It is 4 interlocking hydrocarbons, okay, to form a steroid. Cholesterol is the basis for all steroids formed in the body. Different steroids have different groups attaching to the four ring structure. So the different groups is this area over here that you would have something different attached to it. And then we have eicosanoids. These are derived from fatty acids, arachidonic acid. The most important ones are prostaglandins. They play a role in blood clotting, inflammation, labor contractions, and so on. So these, it's just a little information on that, but this is not too much. Don't spend too much time on this studying. All right, next is proteins. Proteins are what comprise 10 to 30% of your cell mass. These contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur and phosphate. They vary in function by the molecules. So if it changes structure, it changes its function. So, amino acids and peptide bonds. To the left over here, or to the right actually, is a carbon atom single bonded to an amine group. Okay, so here's the amine group, the N and two H's. To the right is a carbon single bonded to an acid group, which consists of a carbon double bonded to an oxygen and single bonded to a hydroxyl group. So we have carbon in the middle, an acid group on one side, an amine group on the other side. So proteins are polymers of amino acid monomers held together by covalent bonds called peptide bonds, and therefore called peptides. Polypeptides are 10 or more bonded amino acids. Proteins are 50 or more amino acids. There's 20 types of amino acids in our body. They contain the amino group, the carboxyl group. They act as either an acid or a base. And then the only thing that's going to be different is this R group. So each one of the 20 amino acids is going to have a different structure where the R is. All the rest of them are going to be the same around here. Okay. In dehydration synthesis, the acid group here of one amino acid is bonded to the amine group here of another amino acid, which causes a loss of water. So that's why this water is coming out over here. The peptide bond of linked atoms form dipeptides. Okay, so here the linked atoms form this dipeptide over here, which consists of two linked amino acids. So we got an amino acid here and an amino acid here. So once again, dehydration synthesis, it takes two amino acids, puts them together, and you lose a water out of that. Dehydration, you're losing a water. And hydrolysis, going this direction of this, you have peptide bonds that link amino acids together are broken when a molecule of water is added. So if we have these two that are a peptide bond that is grouping these two amino acids together, forming a dipeptide, if you add water to that, it will break those apart and put them back into their amino acids. Okay, so going to the right is going to be dehydration synthesis. Going back to the left is hydrolysis. Let's look at the structural levels. So we know that shape and function of proteins, they're very interrelated. Okay, if the shape changes, the function changes. So we've got four different levels here. The primary level is going to be the linear sequence of amino acids. The secondary is how primary amino acids interact with each other. They can form alpha helix coils resembling a spring, or beta pleated sheets resembling an accordion ribbon. Tertiary is how secondary structures interact, and quaternary is how two or more different polypeptides interact with each other. Let's take a look at the pictures. All right, so here it's showing you a primary structure where the sequence of amino acids form a polypeptide chain. So here we have amino acid, amino acid. It's just a bunch of different amino acids forming a chain with each other. So that would be a primary structure. Secondary structure is the primary chain forms spirals. Okay, so now it's forming these spirals. It can also do pleated sheets where it kind of, the shape takes on like an up, down, up, down. Okay, the alpha helix, this is the primary chain coiled to form a spiral structure, stabilized by the hydrogen ions in there. The pleated sheets are primary chains or zigzags. Forming a pleated sheets, adjacent strands are held together by hydrogen bonds. Okay, so these two, they're going to be held together so that they don't go too far from each other with those hydrogen bonds. All right, tertiary structure, which is superimposed on its secondary structure, the alpha helices and beta pleated sheets are folded up to form a compact globular molecule which is held together by intermolecular bonds. So this situation here, this picture is showing you the tertiary structure of transthyretin, a protein that transports the thyroid hormone thyroxine in blood and cerebrospinal fluid. All right, so once we get into the protein structure, which is the tertiary, we know shapes of proteins can fall into two categories, fibrous and globular. So this is the fibrous protein. It creates structure. It's strand-like or woven-like. It's water insoluble, so it does not break down with water, and it's pretty stable. This provides mechanical support and strength. So examples of this would be keratin, elastin, collagen, and some of your contractile fibers and cartilage. The other form is globular protein. These are more functional proteins. So these have a compact shape, they're spherical, they are water soluble, and they're very sensitive to environmental changes. These have specific functional regions called active sites. Okay, so this would be an active sites where different things like hormones and enzymes can come in here and attach to it. Okay, protein denaturation. This is when you break bonds in a protein, which causes it to lose its shape. So when a protein is denatured, also called denaturation, globular proteins unfold and lose their functional shape. The fibrous proteins are more stable, active sites become deactivated. This can be caused by decreased pH or increased temperature. Usually these are irreversible in normal conditions, such as this example here. When you take an egg and you put it in the pan, all the white of the egg is very thin and runny and it's clear. Well, when you add heat, the egg will start to form more structure. It will get more white. It changes its color. It becomes a little bit harder, and you cannot undo that by just going the reverse direction. Okay, quaternary structure This is when two or more polypeptide chains, each with its own tertiary structure, combine to form a functional protein. Okay, so two tertiary structures combine to form a functional protein. Okay. Enzymes. Enzymes are those globular proteins, and these act as catalysts. So catalyst is something that will speed up a reaction without getting involved in the reaction. These catalysts lower the energy needed to initiate a chemical reaction. So catalysts basically make any kind of reaction that they're involved with speed up, and it can make it speed up to like millions of reactions per minute. Some characteristics of enzymes. Most of them are functional. Let's see. They're very specific. They act on a specific substrate. Their name usually ends in ACE, ASE, and they're usually named for the reaction they catalyze. So hydroxylases or oxidases, it's what they're causing to speed up. It's according to what they work with. All right, so some clinical information. Each amino acid has a unique three-dimensional shape. Substitution of one or more amino acids may change the shape and the function of the protein. So an example of this would be sickle cell disease. This is when the substitution of valine for glutamic acid on one chain of the hemoglobin molecule causes abnormalities in the red blood cell formation or function. Okay, I'm going to skip that. I already talked about that. All right, so how does an enzyme actually work? An enzyme lowers the activation energy, which is needed for the chemical reaction to take place. So they kind of prime the reaction. So they allow the reactions to go quickly at body temperatures. So it doesn't need to heat up everything to get it to go. The enzymes allow it to go at the body temperature that's stable. So there's three steps. First, the substrate binds to the enzyme's active state, which temporarily forms an enzyme-substrate complex. Then the complex undergoes rearrangement of the substrate, resulting in the final product, and then the product is released from the enzyme. So let's look at a picture of that. So let's look at the mechanism for enzymes. So here we have the enzyme. It has an active site, which will allow substrates that fit the shape here. So if these two substrates, amino acids, fit in here, they will actually slide right into here, just like these two did, and they will form an enzyme substrate complex. Once they're in there, the ES complex undergoes internal rearrangement. So what that does is a bond is formed between the two amino acids that gives off a water. And then after that, now this does not fit in there any longer because it has rearranged. So the enzyme will throw it out, and when it comes out, it's now the new rearrangement that was from here. So that formed a peptide bond. So now we have two amino acids connected by a peptide bond. And then after the enzyme, it returns to its normal shape, and now it's looking for more amino acids to fit in there. And then this would just continue going over and over again. Anytime amino acids came through, they could form or bond to this and do another peptide bond again. All right, nucleic acids. So these are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. These are the largest molecules in our bodies. They are nucleic acid polymers that are made up of monomers called nucleotides. So if you get multiple nucleotides and you add them together, you would get a nucleic acid. So the nucleotides are composed of a nitrogen base, a pentose sugar, and a phosphate group. There's two major classes, DNA and RNA. So deoxyribonucleic acid and ribonucleic acid. So DNA, there is DNA in every one of our cells. And if you actually unwind a DNA, it's about 6 feet long. So it is jam-packed in our bodies. So our DNA is what holds our blueprint or our roadmap for the synthesis of all proteins in our body. It is a double-stranded helix or a double helix located in the nucleus of all of our cells. The nucleotides contain deoxyribose sugar, a phosphate group, and one of 4 nitrogenous bases. So you're going to see a picture next that has 2 rings. Those are called purines, and that holds adenine and guanine. Where there's 1 ring, that's going to be a pyrimidine, which is going to hold cytosine and thymine. So I'm sure back in high school you took biology and you hopefully remember that certain of these go together and certain of these do not go together. So remember that adenine and thymine go together and cytosine and guanine always go together. So bonding of the nitrogen bases from strand to strand to opposite strand is very specific. So as we said here, we have complementary base pairs. A always pairs with T, and G always pairs with C. And we're going to see that in a picture here. All right, so here we have a nucleotide. Okay, so a nucleotide is formed by having a phosphate group, a sugar, deoxyribose, and a base. In this situation, it's adenine. This other nucleotide here gives a phosphate group, a sugar, and a different base, a thymine. Okay, so this yellow box here is going to represents the T here, thymine, and this green A box is this over here. So when we have on the double helix, you're going to see that adenine always forms a bond with thymine. And these are going to be connected by hydrogen bonds. So anytime you see an A, you're going to be bonding it with a T. Likewise, when you see the C, which is cytosine, it's always going to be bonding with a guanine. Okay, and we have deoxyribose sugars here. This is going to be the ladder that all of these amino acids are connecting to. So that is the structure of the DNA. All right, RNA. RNA links DNA to protein synthesis and is slightly different. First of all, it's a single-stranded linear molecule. And this is mostly outside the nucleus. Okay, so DNA is double-stranded inside the nucleus. RNA is single-stranded outside the nucleus. This contains ribose sugar, not deoxyribose. And in RNA, the thymine is replaced by uracil. So anywhere you would have had a T, which is going to connect to A, uracil is going to connect to the A now. There's 3 varieties. We have messenger RNA or mRNA, transfer RNA or tRNA, and ribosomal RNA or rRNA. Okay, so here's a little chart to kind of put it all into perspective. DNA versus RNA. The major cell site, DNA is going to be in the nucleus. RNA is going to be more in the cytoplasm, outside the nucleus. The major function of DNA, genetic material, directs protein synthesis, replicates itself before cell division. RNA carries out genetic instructions for protein synthesis. DNA structure, double-stranded helix. RNA is a single-stranded, straight or folded. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. So that's why we get RNA ribose and DNA deoxyribose. The bases that are going to form the rungs inside that ladder is going to be adenine, guanine, cytosine, and thymine for DNA. And in RNA, you have adenine, guanine, cytosine, and uracil in RNA. All right, last but not least is ATP. This is all the chemical energy that we get in our bodies. It's like gasoline to power our bodies. This is released when glucose is broken down and captured in ATP. This directly powers the chemical reactions in our cells. It's immediate, it gives us usable energy, and the structure is adenine containing RNA nucleotides with 2 additional phosphate groups. So let's take a look. So here we have a ribose, an adenine, and then we have phosphate groups. So this area here is going to be your adenosine. Okay. If you have the adenine, the ribose, and 1 phosphate group, so like here, that's adenosine monophosphate. So mono is 1 phosphate. If we have 2 phosphates, adenosine diphosphate, that would bring us to here. So we would have 2 phosphates, just these 2, not this one, with the ribose and the adenine. Okay, so that would be there. If we have all 3 phosphates like what's here, we have adenosine triphosphate. And that's mostly how you're going to find it, is the triphosphate. That's the most common form. All right, so the terminal phosphate group of ATP can be transferred to other compounds that can use energy stored in phosphate bond to do work. Okay, so here we have adenosine triphosphate, so that's the one with the 3 phosphates. If you add water to it, it will lose a phosphate group. So that will leave you with adenosine diphosphate plus a phosphate group on its own, which will give off energy. Okay, so loss of a phosphate group converts ATP to ADP. Loss of a second phosphate group converts ADP to AMP. All right, that's the end of our slideshow. Good news is there's no more chemistry, new chemistry at least, in Bio 137 or 139. The bad news is you have to understand this chemistry. So if you do not understand, please ask questions to me, or you can look at YouTube, you can look at Khan Academy, you can look in the Pearson. Plus, they have more videos you can look at. Please make sure you understand this. Okay, see you next video.